So swine dysentery is a disease that is widespread, it is found worldwide, and it has a high economic impact. It is caused by Brachyspira Brachy means short, spira means spiral, and hyo means pig, and dysentery means intestines. It is a part of the phylum Spirochaetosis. It is a gram-negative spiral bacteria. For the bacteria to have this spiral shape, there is a special structure inside the bacteria called a periplasmic flagella. It has axial filaments, which allow elastic movements within the bacteria. This special structure, as a result, gives Brachyspira very bad resistance. Hemolysin is the virulence factor, and it's a strict anaerobe. It cannot exist with oxygen. And it's also a facultative bacteria. For the epidemiology, there are 11 serotypes, and it spreads horizontally in the feces. There is low resistance, but it can survive in the feces. This is because the feces can cause an anaerobic environment. The main host is swine. However, it can live in koi poo, also known as nutria, as well as dogs and mice and rats. These animals will remain carriers for the rest of their lives. The pathogenesis is very unique. It happens in grower pigs. The bacteria does not adhere to the large intestinal wall. However, it moves into the mucus-producing layer of the large intestines, and it damages the epithelial cells, and it causes the increase of mucus production. This will cause very watery mucoid diarrhea. But the bacteria also causes dilation of the blood vessels, and this leads to hyperemia and bloody diarrhea, and it can also lead to necrosis. So in the beginning we have very mucoidy, watery diarrhea, and then it becomes bloody and mucousy, and then we have necrosis. In this disease, we only see lesions in the large intestine as well as the stomach. It also causes gastritis. In the fattening pigs, they lose a ton of weight and they have very grey, mucousy, bloody diarrhea. For the diagnosis, due to the bacteria being strictly anaerobic, we need an anaerobic culture. We can also do PCR, and ELISA is not preferred as there isn't a huge amount of antibodies in the body against it. And for the treatment, give Tylosin, Tilvelosin, and Valimulin, and there is no vaccination. The next disease is intestinal spirochaetes of swine. It is caused by Brachyspira pylosecoli. It is part of the normal gastrointestinal flora, and it is shed in the feces. It is a milder disease than swine dysentery. The hosts are pigs, rodents, dogs, and horses. The pathogenesis is the same. It gets into the large intestines, and then we will have extra mucus production as the bacteria enters the goblet cells, which then leads to mucoid, watery diarrhea. However, the most important thing is the difference between Brachyspira pylosecoli and Brachyspira high dysentery is Brachyspira pylosecoli does not have blood in the diarrhea, and there's no necrosis of the large intestine. However, in the pathology, we can see inflammation and a thickening and reddening of the large intestine, but there's no necrosis. And for this reason, the diarrhea has a grey appearance. But the animals also lose a lot of weight, just like in swine dysentery. And since Brachyspira is a strict anaerobe, for culturing you need strict anaerobic conditions. And for treatment, have Tylosin, Tilvelosin, and Valimulin. And you can also decrease the feed, which allows the gut to have time to heal. And just like swine dysentery, there is no vaccination. The last disease is intestinal spirochaetes of birds. It is caused by Brachyspira alvini. This is the most common. However, sometimes it can also be caused by Brachyspira high dysentery, Brachyspira pylosecoli, and Brachyspira intermedia. The host range are geese and ducks under 5 weeks old, and hens and turkeys in the second half of the laying period, so the hens and turkeys are older. Just like the rest of the Brachyspira bacteria, it requires predisposing factors as they are facultative pathogens. The bacteria is shed in the feces and it can survive there in anaerobic conditions. For the pathogenesis, it lives in the large intestines and it can cause inflammation and the increase of mucus production. In the clinical signs, we will see diarrhea. It'll be watery and mucoid, and they can also have blood in it, so it'll be greyish and red. There will also be decreased egg production and decreased weight gain, and the birds can also get arthritis, as well as pododermatitis on their feet. In the pathology, we can see reddening of the cecum and the large intestines. There can be fibrin and necrosis, as well as arthritis. And the birds can also get nephropathy. When diagnosing, we use a strict anaerobe culture, as it's an anaerobic bacteria, and we can also use gem sustaining, PCR and IF. And for the treatment, give tilazine, talvolocine, and valmuline.